You know that I'm not at liberty to reveal the asset's genetic makeup. Modified animals are known to be unpredictable.
Just kill people, Henry.
That's unfortunate.
What purpose could we have for a dinosaur that can camouflage?
Cuttlefish genes were added to help her withstand an accelerated growth rate. Cuttlefish have chromatophores that allow the skin to change color.
It hid from thermal technology.
Really?
How is that possible?
Tree frogs can modulate their infrared output. We use strands from their DNA to adapt her to a tropical climate, but I never imagined...
Who authorized you to do this?
You did. Bigger. Scarier? Cooler, I believe is the word that you used in your memo.
Hi Year 10, and I'll explain why we started with a Jurassic World clip in just a second, but let's first do our 6 questions, the first one of which is, what is meant by selective breeding? 2, define phenotype. 3, what are different versions of the same gene known as? 4, how do we separate an insoluble from a solution? 5, define an enzyme. And then finally 6, fully explain the shape of this curve we can see here. Good, so our answers then. So selective breeding is breeding animals, plants for their desired characteristics. So not the best characteristics, the most desirable ones. Phenotype is the displayed characteristics of a particular organism. Different versions of the same gene are known as alleles, so you can have 2 alleles, they can either be the same, homozygous, or different, heterozygous. You remove an insoluble salt from solution using filtration. An enzyme is a biological catalyst. And then finally this curve that we looked at, every bit of the curve that goes up, that is where the temperature is increasing as you're heating your substance. When it flattens out as it did, that is because the state change is occurring, and so the temperature stays the same as the state change occurs, and you'd get marks for saying which each of those state changes are. Good. So last lessons we looked at selective breeding and how we can change an organism over time by choosing the characteristics that we want the most and promoting them, so forcing that organism, that animal, with the most desirable characteristics to breed, and so you get that desired characteristic more often. But you are limited in the amount of characteristics or how specific you can get with that process, because you only get the characteristics that have occurred naturally, that have occurred through a mutation. Today we're looking at genetic modification or genetic engineering, which is where you can add any characteristic that you want to a species. And it's a little bit science fiction, it's a little bit out in the future, which is why we started with that Jurassic World clip. In Jurassic World they make the big scary dinosaur by mixing the genetics of lots of different other animals, so a T-Rex, a velociraptor, a cuttlefish and a frog, I think, are the 4 animals. I know all this because it was my brother's favourite film when he was little, and I think I've seen it about 80 times. So in Jurassic World they splice all those organisms together for all their desired characteristics to end up with the one animal that they want. And this is a little bit off in the future, it's a little bit science fiction, but genetically engineered animals are here already. And we can see now this example of a glowfish. This is a fish that has been genetically engineered to glow in the dark. It was originally thought up as a way of seeing if there was pollution in the oceans, the idea being it would glow when there was pollution. It didn't really work, they ended up just glowing all the time, so we just sold them instead. And what we need to know today is how genetic modification takes place. So you can think of genetic modification a little bit like one of those ransom notes you see in films where someone's cut out letters from a magazine and stuck it down to make a new message. Genetic modification requires us to cut up DNA and reapply it to make a new message, to make a new organism with some different characteristics. But to do this, we need a vector, something to stick all that information down on. In my ransom note, that's a piece of paper. You change your message on that paper and it carries it for you. In genetic modification, it's a bacteria cell. That's what we use to carry my new genetic information from one place to another. So we say that bacteria is the vector for genetic modification. It's how we move my desired gene from one place to another. And we'll take a little look now at exactly how we carry out that entire process of genetic modification. So genetic modification is all about getting a desired gene from a particular cell and putting it into a different cell. And the way we do this is via a vector, which is a bacteria cell. And we use bacteria because its DNA is stored in a plasmid, which is the little red thing I've drawn there. So first of all, we go about getting our regular cell and our bacteria cell, and then taking our DNA out of our regular cell. And this is a fairly easy process that we have done in class before. We've extracted DNA from a strawberry. Okay, so it's nice and easy to get that DNA out of my regular cell, and then we go ahead and we do the same thing with my bacteria cell. We remove the plasmid from that cell as well, so that we've got my DNA and we've got my plasmid separate and out of our cell. And then it's about cutting loose the gene for my desired characteristic. So that might be the gene for glowing in the dark, with my fish example, or it might be a gene for insulin production, so that we can make insulin. And we do this by using a special enzyme which works just like a pair of scissors, called a restriction enzyme. And we use those scissors to cut loose the desired gene, but also to cut a gap within my plasmid. We use the same enzyme so that that hole is the perfect size for my desired gene to sit within. Okay, and by using that same pair of scissors, that same restriction enzyme, that plasmid is left with something called sticky ends, which is a key word and a technical term, albeit a ridiculous one. That means that the hole is the perfect fit for that desired gene. It's sticky, and so when it comes to the next bit, which is putting that desired gene into that gap, it fits perfectly. So my desired gene gets put into that plasmid, and then it's stuck down using a ligase enzyme, which is works just like the glue to stick that down in its place. So thinking of my ransom note example, this is like the glue sticking it down into the plasmid, which is the vector, the paper, okay, having used the scissors to cut everything up. Okay, and here is a bottle of glue if you're unsure what that looks like. So now that I've got my altered plasmid, that's the difficult bit done. I've stuck my desired gene into my plasmid, and now it's just a case of sticking that altered plasmid back into my bacteria. And this bit is why we use bacteria, because once my plasmid is within the bacteria, the key important thing about bacteria is that it multiplies mega quickly. So now I don't just have one of those altered plasmids, I've got lots and lots of them. And so I can start to use that to genetically modify other things, because this bacteria can be inserted into and can infect other organisms, and it will transfer that altered gene, that altered bit of genetic information into it too, if we do it properly. Okay, so we've got lots of these genetically modified plasmids to play with now, and it's important to have lots because it doesn't always work. Good. Within that process there are quite a few key words, and it's really important that you learn that process of genetic modification and all the key words, the different enzymes, the vector, all those words that go with it, which we'll repeat lots of. but it's important because it comes up an awful lot in exam questions like the one that we're just about to have a look at now. So this is a really common example of a question asking you about genetic engineering, and it's how it's carried out. It shows you this diagram about how it takes place, and it's far more complicated than anything we've looked at. It then says enzymes are used to genetically engineer the bacteria. Write down the name of the 2 enzymes used and explain what they do. So for 4 marks, 2 marks come from naming those 2 enzymes, 2 marks from saying what they do. So first point, restriction enzyme. You get a mark for just writing that. And then we'll say what it does is used to cut free desired gene and to cut hole in plasmid. So 2 marks for that there. Your other 2 marks is for saying ligase enzyme used to stick desired gene into plasmid. Okay, nice and simple, easy form. That's just about it for this lesson. The next lesson is pretty much a straight continuation from this, so it's less 2 lessons and more part 1, part 2. So go ahead and watch that now. Have a great week. Bye bye.